So I'm here today to talk to you about strategies towards improving the utility of your big data. And if you don't already have big data, you might have it soon. Or you may be consumers of it and may not really understand how it works or how you might be able to take better advantage of it. Some of you may have multiple sets of big data and you may not have originated it, but you need to use it. So where I work is part of the U.S. National Library of Medicine, part of the National Institutes of Health. And it really has a broad mandate for providing information for you all, everything from consumer health type information all the way to aspects that I work on, which is research-based chemical information, chemical biology type data. So I work at the U.S. National Center for Biotechnology Information, NCBI, which houses things like PubMed, which has like about 1.5 to 2 million users a day. They serve terabytes of data. They have genes and DNA sequences, proteins, but they also have a resource that I work at called PubChem. So whenever there are genomes and whatnot published, they go into NCBI, and we annotate and cross-annotate the same information. I'm not really going to talk to you about PubChem, but just to let you know, this is our website if you're interested to learn more about the project based on some of the things that you learned about today. But I will mention that what we're trying to do is to bring together big data that I care about, which is things on the chemical information side, and so that we know what chemical substances are and what their biological health effects are. And these aren't just drugs; these are things like diesel fuel or the bug spray that you put on yourself. The chemicals inside. What do they do, and how are they important to you? And just to give you some idea of what our big data looks like, and I don't really expect you to look at all these different graphs, but just give you some idea of some of the basic data that we have. How has it grown over the course of 10 years? So in terms of people who give us information. So PubChem is an archive, and so we depend completely on people to push data into this archive. But as a part of that, we have a very large number of chemical substances that are being stored in there. We have on the order of about 125 million chemical substance descriptions. Of those, about 50 million of them you might consider to be chemicals that you would think of as being things like aspirin or other things like vinegar, acetic acid, and so on. But it's really grown as a function of time as we've been around for 10 years and people caught wind of this and started adding data in. We sort of created our own big data problem, and we had to solve a lot of really basic problems about how to deal with this information. One of the first things we learned is that, you notice that all data has errors, and, I would tell you that big data has big errors. And just to give you some ideas of what we're talking about here is, imagine that you have an error rate of 1 in a million, which is to say that you have 99.999% data accuracy. And that's really a very good data accuracy if you were to have this. If you have 1 trillion facts, you have 1 million errors. Can you deal with a magnitude of 1 million errors inside your data? I will tell you that as an archive, and for various reasons which I won't get into, we probably only have 1 or 2 nines in our information. It's not that the information is just completely erroneous. It's that you can get inconsistencies when you start to combine large data together based on what people think about their information and what they provided to you. It's to different degrees of granularity. It gives you different aspects of the information that may or may not be relevant to you and your use case. So what do you do to sort of help stamp down and get as many nines of data accuracy that you can? You can try manual curation, and people talk a lot about curation, but there are limits about what you have. And in fact, many of the resources that are put inside our archive is curated. They've already done curation. But what you find is that, it has its limits. We're human, we make mistakes. And you may have a record, that record may have tens or hundreds of facts inside that record. Some percentage of them are going to be wrong. because somebody forgot to dot an I or cross a T somewhere. Or they capitalized something that they shouldn't have, and suddenly it becomes something else. So what do you do? So what I'm going to go through is a series of strategies to try and improve, to sort of demonstrate ways that you can improve your error, or say eliminate the error or somehow clamp down on it, but also ways that you can structure the information that you have left. And I'll just go through these really quickly, and then I'll give you examples of each. One thing you need to do is you need to understand your information that you have. Make sure you understand the scope of what you have. You need to do some type of data normalization to your data. If you just take it as is, you may be very unhappy for it. You need to trust your data, but you better cross-check it to make sure that it's what you think it is. Do various types of filtering to try and make sure that the information is consistent for the use cases that you have in mind, because your data may cover other use cases that go beyond the scope of what you're interested in doing, which has something to do with normalization as well. And the other thing that we've noticed is these error feedback loops, where errors propagate. Somebody found an error, you went to a website, you got some information, you read it, great, you read a paper, but there was an error in that paper that they may have gone back and they've corrected. But now you put inside your system the erroneous data, and meanwhile someone else found data inside your system, put it inside their system, and then we end up getting all this data back. Some of it's been corrected, some of it hasn't been corrected. It may have been corrected at a particular moment in time, or it may reflect the errors that were in the actual original document, and you want to preserve that in certain cases. But as a process of doing that, you may be pushing out erroneous information for people who don't know and aren't in the know. You wouldn't know what to say. Oh, well, this one's telling me it's A, and this one's telling me it's B. Oh, yeah, but we already knew that A is B, but we have to keep it this way because this is what the original publication said. Okay, so known unknowns. There's this guy named Donald Rumsfeld, and he had this thing, there are known knowns, there are things that we know, but there are known unknowns, which is to say that there are some things we do not know. We know that we don't know. But there are also unknown unknowns and things we don't know what to do about it. Well, in the case of chemical space, we have to know our information and to see, well, what are our known knowns and known unknowns, and try and get a handle on what are some of the unknown unknowns. And chemical information is it's complex. You have a particular chemical structure, but you can take that same chemical structure and you can draw it in a myriad of different ways, because they have these things called tautomers and resonance forms and whatnot. But all these things that I'm showing you here are all guanine, but these are different ways that we see the same thing. But knowing that we have this issue, we can start to think about how we might be able to correct or do something about it. In chemistry, structures they can have multiple different forms naturally. So here's a natural product, which is from a defense chemical of abdominal glands of a row of beetle species. Well, this natural product can be represented in this form or that form. They're both correct. But in your use case, you might think one's more correct than the other. Some people, when you deal with information, you can represent it in different ways, much like you can in the case of something called sodium acetate. Acetic acid is something you have inside vinegar. If you react it with sodium, you make sodium acetate. Well, some people like to show just acetic acid and the sodium metal, as opposed to having something that's somehow combined together. But when you start to think about this information, if you think these are different, you might have an issue, but if you knew that they're the same, it may be helpful in how you think about the information, how you can combine it later. And then you can take the same information that you say, okay, now I understand it, but now you change the context. Here's the chemical context, and here's the biomedical context. Sodium acetate is a trihydrate. It's a very specific thing to say that it absorbs water, and this is how you would buy it, and so on. But the same thing can have a different meaning as you change the context or your use case. Data normalization improves utility by helping to refine your identification. So now you understand what some of your issues are. You can design some type of a platform, and this is what we do with ours, to clean up our chemical structures, to normalize them. But you can imagine with your information content, you could normalize it so these different representations of the same thing may only have a single form, for example. Or you have other ways that you can collapse down things that are equivalent or near equivalent into something that can help you to stitch together the fabric of your data. So in some ways, you're starting to already add structure to your information by doing these types of normalization steps. Trust but verify. There's this famous guy, back in the 80s, every time he was dealing with the Russians and it came with these ICBM treaties, he would always say, trust but verify, which is actually a Russian proverb. And I'm not going to try to pronounce it. But it's interesting to see that, yes, you have something that an information source that you have, but when do you know when to trust it? If you just trust it blindly, you may be very disappointed. And there's this other famous guy, John Kerry, when dealing with this more recently with the Syria chemical weapons disposal, thought, well, maybe we should change this, not trust but verify and verify. Which is to say that with your information that you have, you better make sure that it's as supreme as you can, but it may require you to go through extensive steps to try and, well, be sure you have what you think you have within the context of your scientific data. So to give you an example for things that are important for PubChem, we took various disease ontologies and vocabularies and did a cross comparison to see, well, how do they compare between their disease concepts? We use MESH as the medical subject heading, which is from the National Library of Medicine, used to index the biomedical literature. It's a series of dictionaries, one of which includes lots of disease concepts. But the idea here is not necessarily to look at all these numbers, but to notice that a lot of these numbers are not 100% or in the 90s. They're in the low 30s or even in the 10s, which is to say that the concepts used for diseases across these various ontologies and things that people use to structure their data, they don't necessarily agree. Which is to say that you either need to mash them up in some way or to somehow disambiguate those things that these concepts describe. They may be describing similar things or related things or completely dissimilar things. And if you don't get a handle on this, when you add structure to your data, which most people are using ontologies and classification systems and vocabularies to modify your data, or say to add structure to your big data, you might find a lot of idiosyncrasies when you go from one vocabulary scheme or concept scheme and go to another. And it may be that you have to do multiple mappings to somehow get a handle on what's going on there. Consistency filtering improves precision by removal of outliers. So if you can imagine, you have some type of a target, and the usual thing that you're really, really hoping for is that you get everything right in the center of the target. Everything is grouped really close together, which is to say it's precise, and it's because in the center target, it's very accurate. Well, you have big data. You don't necessarily understand the accuracy of it per se. However, you can improve the precision. You may not be at the center, but at least you can eliminate the outliers so your data isn't as scattered. By having various means of consistency filtering, you can somehow get a handle on how varied your answers are for the same information. To give you some ideas on the PubChem side, we were looking at various aspects about how we have different structures associated to different chemical concepts found within various types of vocabularies, and designing various ways that we can minimize the spread to say that multiple chemicals are being referred to by the same concept. Sometimes chemical names can be very promiscuous. DNA, we all know what DNA is, right? Dinitroaniline. In this case, one could say, okay, I think of DNA as one thing in one context, but it could mean 1, 2, 3, or 4 other things as well. And you have to be careful when you do a matchup to your vocabularies to make sure that they're actually dealing with the same information. But really the take-home message for us is that we were able to eliminate, for the same queries, we're basically able to pick a chemical, or at least to find one that's consistent between our contributors, and to get some sort of a crowdsource idea of what's going on with this information. So the idea here is we can keep the consensus and get rid of the rest. We don't eliminate it. In fact, we keep all of the original information. As an archive, we preserve all of the data. We don't get rid of anything. However, we can hide a lot of the information by default. But that we can also do is to focus the information so that when you query, say for aspirin, you don't see Tylenol, because you have aspirin-free Tylenol. It has aspirin as a key word in that, and sometimes people get confused and say, oh, well, Tylenol is aspirin as a result of that, because when they search for aspirin, they didn't get what they expected. And they look at the results that come back and say, oh, your database is full of errors. Not necessarily full of errors, it just handled things that you weren't necessarily expecting. And if you're looking to use something for research-based purposes, typically what we would do is just put the most likely answer at the top. But then we just have some sort of a down-weighted set of results as things become, well, less specific. We found that was not what our users expected. They expect to see the correct answer and just the correct answer and nothing else. But you might find the same thing too, is that if you start to harness big data, you might find you query something and you get lots of answers for the same question. But it doesn't necessarily mean that they're wrong. It could simply mean that you haven't normalized and filtered down your information to the point where, for your use cases, you're happy with the answers you get back. And that can be difficult to do. In the case of chemicals, there are lots of things that can go wrong, in part because you might draw your chemical structure and it could be absolutely correct. But then you save it, and you save it into a format, but then you load it back into your software again, and may or may not be exactly the same way that you had it before. So when you move data around between different archives and resources, not just chemical information, but data in general, every time you touch it with an algorithm or import or export it from a software package, you may or may not have the exact same thing that you started with. In fact, we could start to see patterns in what software, even software versions, that were being used when people were giving us data from various sources. We even invented a term called robochemistry to describe what's going on, in that every time algorithms were touching the chemical structures and doing some type of normalization, in some cases they were horribly corrupting the structures, and being able to reconstitute them in some way became somewhat problematic. But then, people were saying, oh, well, your structures are wrong inside your system. But they weren't necessarily wrong, it's that they got corrupted. Even curated information is getting corrupted when these types of things happen. So you have to be very careful with how you handle the information, whether you're a downstream user or the one creating these types of archives, because strange things happen when you apply software to data. And even as you structure the data inside the database, you may change its meaning. So you have to be very careful. Addressing feedback loops. Well, one thing that we sort of came up with is we started to create white lists, gray lists, and black lists. So a white list is to say, I am absolutely perfectly happy with, I know that this thing is that. And you set it in stone. It can only be that. Because we do various types of things with filtering and normalization, it's possible that as a function of time, the correct answer becomes wrong, because someone gives you the same information erroneously multiple times. And if you're using various types of cleanup algorithms, the correct answer, the signal gets drowned out for the noise. Well, one way to address that is by saying, oh, it can only be this. But in some cases, you can only narrow it down. You can narrow it down to a list of candidates because you know it can't be anything else but those things. And you can then apply your consistency rules based on that. And I've already shown you lots of different ways chemical structures can be represented, but the same thing is true for sequences or other types of scientific data. It could also be in the case of physical properties or spectra or other things like that, where You might not have the exact same thing, but it's close to it, or it has to be one of these things. And if all else fails, you can create is-not lists. And this is typically what we do to fix errors quick, which is to say that this thing cannot be that. But really, the errors of what it cannot be is infinite. So really, this is not a winning strategy to say, oh, okay, I'm going to say it's not that, because someone else can create some new error tomorrow and you won't filter that one out. It's better in many cases to triage things down to say, well, what can it be? The other thing that we've really tried to do is to prevent error proliferation at the data source. When people give us information, we subject it to all types of analyses, and we'll tell them when they're making things worse. We've also have engaged and worked with a lot of manual curation efforts that exist worldwide. In fact, there are governments that fund various types of projects that do manual curation. These are tens of people, students oftentimes, paid to curate information. But when I cross-compare this curated information, I often find errors, human errors, silly errors, or most times they do a great job. But I find that when I help to combine these together and then work with these, I get a positive feedback loop in that they start to converge on the proper answers. But all I can do is say, look, you're inconsistent. Are you correct? And oftentimes we get the feedback that in some cases there are some real disagreements as to what the real structures are or what the data is. But oftentimes it starts a dialogue that those communities can work on and fix. And I can stay out of it for the most part. And they can do the parts that need to be done. But really, if you can, with your big data or your big data sources, if you can let them know what's wrong and help them to mitigate the errors, the next time you get an update, maybe these problems go away for you. Because oftentimes they're experts in these areas and they're better situated and oftentimes maybe better funded to correct these types of issues than you are. Because if you just fix it for yourself, anybody else who uses this data, and then they copy it and then put it into their resource, and then you start to use them, you start to see the same error over there. So oftentimes it's better to, I think, kill the error at the source when it can be identified. So in terms of my top 5 or 5 quick error suppression strategies, identify quality, understand your information, the known knowns, the known unknowns, and so on. Normalize your information. Trust your data, but verify it. Cross-compare it. Work with your various types of curated sources, and if you don't have multiple sources, get them. So you can get some second opinions or third opinions about what things are. The more opinions, the better, in my mind. Consistency filtering can help a whole lot. Oftentimes, it removes a lot of the fuzz in the information that you have. And if you don't do this, you might find that you get many more answers back in your analyses than if you do some type of a filtering down. You can always expose this information out afterwards, after you do your analysis, about the other equivalent forms or near forms that may be of use. And the last, definitely address your error feedback loops, especially if you're dealing with multiple data sources. All right, now what? You clean up your data, but it's huge, unwieldy, unstructured. How can it be made more useful? Well, one way is cross-link and annotate your data, provide a better context and identify associated concepts. Establish similarity schemes so you can find near neighbors and related records. Associate things to concept hierarchies wherever and however you can to help you navigate between records. Perform various types of data reduction to suppress your redundant information. Oftentimes you're going to find you have lots of the same thing again and again. And last, find ways to be succinct, basically by hiding a lot of the details that's available. To give you some ideas, what we do inside PubChem is, we have information about this substance is somehow related to a protein. We can start to tell you things, well, it inhibits this protein. Or it's mentioned inside of this particular publication. This gene is somehow expresses this protein, and so on. We can start to expose these types of cross-links. Otherwise, we just have simply bags of substances, bags of proteins, and links between things without any context. I'm running out of time here. But the other thing about establishing similarity schemes, it's kind of like the Russian dolls, right? A doll inside a doll inside a doll. And if you have some way of hierarchy to express information, you can sort of zoom in or zoom out on your information data. We harness this for chemical records by allowing to find similar related information rapidly. By associating things to concept hierarchies, you have a hierarchy. You can associate chemicals, proteins, genes, patents, publications, whatever you want to it. It's a way to navigate the information that's there. Well, we do this inside PubChem. We can tell you things about a particular chemical or entity or target based on these hierarchies. But it also allows us to allow you to browse our entire contents using them or to mine them. The last point is really two different points, and it really comes down to how you can reduce your data down into simple statements. In this case, we're using linked data or RDF. You can say something on the orders of atorvastatin may treat hypocholestemia. You have some type of a subject, predicate, object trio, but you also have provenance information. If you reduce your data down to these types of statements, it can really help. But if you want to be succinct, if you leave out the provenance information, it can really simplify things a lot, because we find that we get the same information again and again, and a lot of it we simply tell you, well, where did it come from? Who gave it to us? Where did they get their information, and so on. So again, a top 5 for ways you can organize your data. And just give you some concluding remarks. For scientific big data, it contains an amazing amount of information. I mean, really groundbreaking stuff that allows you to have opportunities to make new discoveries. If you just simply knew to ask the right questions, if the data is organized in just the right way, you can connect the dots and make true discoveries. It's already there. It can benefit from strategies to massage this data, and PubChem is trying to do its part in this area, at least within the space that we are in chemical biology. We're trying to make the chemical data very broadly accessible to you so that you can make more use of it. In the case of Wolfram, they could put it inside their alpha system and do more with this type of data. We cross-integrate it to various key scientific resources so you can find other related information. And we're doing our best to sort of suppress their errors and propagation of those errors, but also making it organized so you can take better use of it. PubChem is a project with a number of us working on it, talented group of folks. And if you have any questions, I'd be happy to answer them if there's any time. But if you think of something later, if you don't think of or have time to ask a question now, feel free to email me. Thank you.